For this lesson, what we're going to look at is the anatomy of introduction, or as I like to call it, an introduction to introductions. So we're going to look at most research articles that you'll read will have a general trend about them. And so the trend that you'll see, that you most commonly will see, are what's called the inverted triangle. So most introductions are organized in this way. And so what an inverted triangle means is that at the top of the introduction, the first paragraph, for instance, is going to be the broad relevance of the research. So what broad problem are we looking at here that you can easily relate to usually, okay? And then at the end of the introduction, the authors will typically talk about the specific focus of the study or the purpose. Other things you might find here discussed here are the research questions or even a hypothesis that sort of led the research, the experiments for the research. So in the middle will be a transition between the broad relevance and then the specific focus of the study. And so what will be discussed is what previous work has been done and what is known in the field. Okay, and this will be a lot of citations of previous research articles typically. And what may also be discussed is if there are conflicting studies, for instance, if there are two studies that say opposite things, then they may discuss that. And this leads to also another thing that might be discussed is gaps in the field. Okay, and so typically the authors will highlight these gaps because the purpose of the study is to address these knowledge gaps in the field. Okay, so as an example, we're going to look at this paper by Thomas and co-workers. So this is called Protein Nitration is Mediated by Heme and Free Metals through Fenton-type Chemistry, an alternative to the nitric oxide superoxide reaction. Okay, and so the way this is organized is once again you have your title here, you have the authors, their affiliation, and then in bold here is the abstract, and then the introduction follows right after the abstract. So I've color coded this to sort of highlight the organization that the authors chose to use for this introduction. So here's the top of the introduction. So this is where we expect to see the broad relevance. And then here's the bottom of the introduction, just above the materials and methods, where we expect to see the research question, a statement of the research question or the hypothesis. So looking at the first paragraph, again, we expect to see the broad relevance. So what is the broad relevance here? As indicated, what is talked about in this paragraph is nitrated tyrosine residues are a biomarker of several human diseases. Okay, so I'm not going to go into each sentence of this. I've just summarized it here. If you want to look at each of the sentences within this paragraph, I've posted this paper with the color coding on it so that you can read it, okay, on web courses. But the general point of this paragraph is to introduce why nitrated tyrosine residues are important. And so they have consequences in human health disease, or they're biomarkers. So they're interesting from that perspective. All right, so let's go to the end. So this is where we expect to see our hypothesis statement, and in fact there is one that we'll come back to in a sec. Okay, so this is where we expect our hypothesis statement and purpose of the research. But how do we get from point A to point B? So from here, this gives the broad relevance again. The first sentence of the second paragraph hints at the purpose of the paper. So the sentence, I'll read this sentence, says several mechanisms for nitrated tyrosine formation have been proposed, and its origin remains controversial. So the fact that it says that there are several mechanisms and they are controversial, that indicates there's an argument in the scientific community about how this mechanism happens. And so it leads to the thought, so it hints that the mechanisms of NT formation are going to be discussed within this paper. That is the purpose of the paper. So in what follows in this paragraph is some more discussion of the previous work, particularly in this case, looking at this molecule called peroxynitrite. And so the peroxynitrite is known to cause nitrated tyrosine residues, and so they're discussing briefly some of the work that's been done. In the third paragraph, they're also adding to this discussion that there's an additional uncertainty. Other studies indicate that peroxynitrite is not necessary for tyrosine nitration. So that's interesting. So that gives a second, completely different mechanism by a completely different pathway in which nitrated tyrosine nitration may occur. And so that indicates there are at least two competing pathways in the literature. And they're probably, again, this hints at what the purpose of the paper is to to provide data that either helps indicate that there either both of these pathways occur or maybe just one or the other. Okay. Finally, here's the final paragraph where we expect our hypothesis and statement. So, and purpose. The top paragraph, or the top part of the paragraph, the first two sentences say NT has been shown to occur on distinct proteins rather than in an indiscriminate manner. This may result from variations in protein susceptibility to nitration due to conformational differences or to association with redox metals. So this hypothesis statement is somewhat hidden, and this may happen. You may notice this as you read papers, is that no one will sit there and say, we hypothesize that this and this will occur. Keywords that you may find instead are, for instance, this may result. So that's not a statement of absolute certainty. You can read it as sort of a guess, and that is basically what a hypothesis is. Other words you may see are suggest is a very common one as well. But what the hypothesis in here is redox active metals can contribute to tyrosine nitration. Okay, so that's the hypothesis statement. That's the hypothesis that they will be testing in this paper. And the third sentence is a short description of the work that follows, and it can be seen as the purpose of the study. So we evaluated the relative efficacy of the synthetic peroxynitrite NO superoxide cogeneration, peroxidase, and redox active metal pathways to promote NT formation. So that is the goal, is to evaluate how peroxynitrite, or a different way of making peroxynitrite with nitric oxide or superoxide, or peroxidase, or other redox active metal pathways, all work together to form NT formation. And so the overall purpose of this is to see how these different components interact in order to do this reaction.